Hello, everyone, and thank you for joining us today. Today's webinar is the Plant Physiology webinar on the topic of hypoxia in plants. And we have four speakers, Bart, Amanda, Moto, Beatrice, and we have a host, Rashmi, and we have a moderator, Alicia. And Alicia will be in charge of introducing everyone as we go along. We're celebrating today the January 2025 Plant Physiology focus issue on hypoxia in plants. It's a fantastic focus issue, and I will put the link to it in the chat in a few moments. It's online now. This webinar will be recorded, and we will post the recording on YouTube. You can find all the Plant Physiology webinars that we've recorded on the Plante YouTube channel. We'd like to make a special thank you to ASPB members whose membership dues support this project. If you'd like to join ASPB, you can use the code PRESENTS10 to get 10% off your membership dues. I would also like to invite you to consider attending the plant biology meeting in Milwaukee, Wisconsin this coming July. The plant biology meeting is a big meeting. It's very diverse, and it has wonderful opportunities for great science and networking. So have a look at the website and see if you can join us. Alicia will have the opportunity to ask a few questions to the speakers, and so please type your questions into the questions box. If you have any issues, you can email me. I also want to let you know if you need a certificate of attendance, you will get an email tomorrow with information about how to attend, how to get that certificate of attendance. So I will now introduce Alicia, who will then take over the webinar. Alicia is a postdoc in Professor Pierdomenico Perata's lab at the Santa Ana School of Advanced Studies in Pisa, Italy. Her research focuses on deciphering the molecular mechanisms integrating hypoxia signaling with sugar availability, using Arabidopsis thaliana as a model organism. Alicia is interested in fine-tuning of ERF7 transcription activity by post-transcriptional modifications. Over to you, Alicia.
Thank you, Mary. I would like to welcome our speakers and everyone tuning in today. Thank you for joining us. Today's seminar is hosted by Rashmi Sasidharan, who is a professor of plant stress resilience at Utrecht University. She studies how plants sense, respond, and acclimate to flooding. She combines genomics, genetics, and molecular physiology to probe the roles of the associated stress signals, so not only hypoxia, but also ethylene, starvation, and reactive oxygen species. Rashmi is also interested in identifying the signaling networks, facilitating flooding recovery upon reoxygenation, and exploring the parallels with drought. Unfortunately, Rashmi couldn't join us in person today, but she sent us a pre-recorded video, which you can already see, and we will play it for you now.
Hi everyone, it's my pleasure and honor to introduce this special issue on plants and hypoxia. The star of our focus issue is the molecule oxygen, or rather its limited availability, which causes hypoxia. And plant hypoxia has mostly been associated with flooding, but as you can see on this cover image, it also happens in other instances, including high elevations and during microbial interactions. Hypoxic niches can also exist within the plant body due to specific physiological and anatomical traits, such as in tubers, meristems, and fruits. The mechanistic understanding of hypoxia as a signal influencing plant growth and development was largely investigated in the context of plant flooding responses. That, however, has changed dramatically in the last 15 years or so, spurred by the discovery of the plant cysteine oxidase end-day-grown pathway of oxygen sensing. And while this pathway remains a focal point for hypoxia research, two questions have driven current research in the field. And now I take this from our editorial for this special issue. So the first question is, how is oxygen sensing integrated with other signals to modulate plant acclimation responses? And secondly, how does the broader environmental and development context shape hypoxia responses and the adaptive traits of plant cells? So our focus issue has several research articles, 9 updates, and a topical review, all of them providing an overview of the latest developments in the field. And what I would like to do now is quickly take you through the updates and reviews published in this special collection, starting with this one from the labs of Emily Flashman and Oscar Lorenzo, providing an overview of the endegron pathway for oxygen sensing and describing what we know currently about the functional and structural features of the components of this pathway. Then we have Daniel Gibbs, Freddie Theodoulou, and Julia Bailey-Serres giving an update of the multi-tiered regulatory networks that control plant responses to hypoxia. Then we have a topical review authored by Bakshi and Gilroy, who give a comprehensive update and historical perspective on the role of calcium signaling in hypoxia responses, while the labs of Sean Hartman, Angelika Mustrov, and Romy Schmidt-Schippers put together collective knowledge on how hypoxia signaling interacts with energy, redox, and hormonal cues. Then we have an update authored by Hans van Veen, Paolo Triozzi, and Elena Loretti, who give us an overview of the metabolic strategies that are used by plants to deal with hypoxia, not just during flooding, but also under non-flooded aerated conditions. And then we have a very nice review from Motoyuki Ashikari, who's also a speaker today, and Julia Bailey-Serres, who discussed the classic escape and quiescence adaptive survival strategies seen in rice. Then we have an update led by Hardy Rolecek and Chiara Pucciariello, who discussed advances in seed hypoxia research, which is also a very important topic from an agronomic perspective. And a relatively new and exciting area of research in our field has been the association of hypoxia with altitude and geography. And this is the topic of the update led by Mike Holsworth and Pierdomenico Perata. Finally, we come to the last two updates. Research in our community has been really accelerated thanks to some very impressive developments in the field related to tools that allow us to not just measure oxygen dynamics, but also visualize them and manipulate oxygen responses. And this is highlighted in the update from Dan Weitz, Ole Pedersen, and Markus Schwarzländer, who write about the latest in oxygen measurement and visualization tools. And finally, in the update from La Villa Puerta and Giuntoli, we have a review of the utility of synthetic biology techniques for hypoxia research. And Beatrice is also one of our speakers today, so we'll hear more about that. And with that, I would like to round up with a word of thanks to my co-editors, the Plant Physiology team, all the authors and reviewers who contributed, Mary and Alicia for organizing and hosting this webinar, and of course our speakers. So to everyone listening, I really hope you will enjoy the webinar. And before I hand it back, I would like to quickly advertise the fact that we are having a plant hypoxia community meeting this year in Japan. Registration is now open and all the details are on this slide. And with that, Alicia, it's back to you. Thank you.
Thank you, Rashmi from the past. And now it's my pleasure to introduce our first speaker, Bart Nicolaï. He is a professor at the Biosystems Department at the University of Leuven in Belgium. Bart's research area is post-harvest biology and technology of fruit and vegetables, with a focus of physiological responses of fruit when stored under hypoxic conditions. His lab also develops techniques for non-destructively measuring quality attributes of fruit and vegetables. Today, he will tell us about his recent publication on hypoxia in tomato fruit during ripening. You have the stage, Bart.
Okay, thank you, Alicia. Let me just share my presentation. We'd like to start with thanking the organizing committee for having me. It's a pleasure and also an honor to share some of our research with all of you. And as Alicja mentioned, my lab is a post-harvest lab, which means that we develop new technologies to extend the storage life of fresh fruit and vegetable based on insights into the physiology of the fruit or the vegetable. And one of the products that we work on a lot is tomato fruit, not only because it is one of the most important horticulture products that are produced in Belgium, but also because it is, of course, a model system for climacteric ripening of fruit. And one of the physiological processes that we are very much interested in is, of course, respiration, because post-harvest is all about reducing the respiration rate. This means that we have done quite some research in the past on gas exchange of all kinds of fruit. And because tomato is a bulky fruit, it has some resistance to gas transport inside the fruit. And together with the fact that these fruit are respiring until the very moment that you consume them, this leads to potentially to hypoxic conditions inside the fruit. So the concentration of oxygen inside the fruit is lower than the ambient concentration. At least this is the hypothesis that we started with. And there's some evidence in other fruit species that this is indeed the case. For example, in the past we have done some work on gas transport in palm fruit, apple and pear fruit. And in this slide, you can see cross-sections of these different fruit of different cultivars. And the color basically denotes the oxygen concentration. And you can see that in some of these cultivars, for example, in Braeburn apple, the concentration is very low, can be even anoxic in the center of the fruit. And this has major consequences because it would cause the fruit to basically switch to fermentation metabolism. But also in the other cultivars, there are oxygen concentration gradients, and there are also carbon dioxide concentration gradients which follow the same path. So what we wanted to do was to find out whether we also have such gradients inside tomato fruit. And this is research that has been carried out by my then PhD student Hui Chao, who is now an associate professor at Shanghai Normal University in China, with Peter Verboven, one of my research managers, and then Shuai Tong and Ole Pedersen of the University of Copenhagen. And the way how we approach this is that we use models that are based on first principle physics. And the reason why we use models for this is that it is not too easy to measure oxygen concentration inside the fruit. And if you want to have like an oxygen concentration profile, you basically need to measure the oxygen concentration at every position where you want to know the oxygen concentration. While with models, you can basically make one prediction of the oxygen concentration or the carbon dioxide concentration in addition to that in the entire fruit. There are also no really, there are some developments in oxygen imaging methods, but this is still kind of an underdeveloped area. Of course, these models then need to be validated, otherwise you cannot really rely on them. And I'm not going to go into the details of the type of models that we are using, but basically oxygen transport is governed by some sort of a conservation law, mass conservation law, which says that the oxygen that is consumed by respiration inside the fruit needs to be transported from the ambient into the fruit by diffusion processes. And the two parts of this equation basically reflect this. So the left part is the diffusion part, which is basically fixed law of diffusion in combination with a mass conservation law. And in the right-hand side, we have the reaction term, which represents respiration. And together, this is called a reaction diffusion model. It's a model that is used a lot, and that is very important in mathematical biology. So how did we solve the model? Well, we started with taking MRI images. MRI is magnetic resonance imaging, which is a medical imaging technique that is very sensitive to water mobility. And because the water mobility in the different tissues of the tomato fruit is very different, it is possible to easily make a distinction between the different tissues inside the tomato. Here we have the pericarp, the septa, then we have the luculi that are typically filled with a gel, and then we have inside the columella, so the central axis, and the ovarium and the seeds that are also inside the luculi. And by using some image processing techniques, it is possible to segment this image and then to visualize it. We actually used false colors to make it a little bit easier to interpret the images. And then we translated this in a so-called finite volume mesh, which is a kind of a requisite for numerically solving the gas transport equation over this geometry, which basically means that we have to subdivide this geometry or the different tissues in this geometry into small volumes that we then that we then use for solving the equations. And we also needed to have the diffusion coefficient, of course, and for this we took with an X-ray machine small images or images of small excised tissue samples, and all the green that you can see here is basically the airspace in between the cells at a resolution of around 4 micrometer. We then translated this image in a pore network representation, which means that we represent every pore in this tissue with a sphere, and all the spheres are connected, and these channels basically represent the connections between the different pores in the tissue. And we used another model in a so-called numerical experiment where we were able to, by establishing a concentration gradient over this volume, to calculate the oxygen and carbon dioxide flow in the tissue. And from that, we could derive a value of the so-called apparent diffusivity, which is a kind of a parameter that describes how fast oxygen or carbon dioxide diffuses inside the tissue. Then the last component of the model was the reaction kinetics. And for this, we did some oxygen depletion experiments where we put small samples of the different tissues into vials and then just waited until the oxygen was consumed. We measured the oxygen decline during the depletion experiment as a function of time. And from that, we were able to establish the reaction kinetics, which were kind of modified Michaelis-Menten kinetics. And in this graph, you can see the kinetics for a mature green tomato. This is the kinetics for orange tomato, and this one is for red ripe tomato. And what you can already see is that there's, of course, an effect of oxygen. If there's no oxygen, there's no respiration. And then it kind of levels off at higher oxygen concentrations. And the oxygen consumption rate, which is in the vertical axis, is the highest in the orange tomatoes. And this is because the climacteric peak as it is highest here. So ripening proceeds the fastest when the tomato is in this particular stage. So at this point, we had all the ingredients for the model, and then we did some simulations. And in these contour graphs, you can see the oxygen concentration profile. So the legend is here at the right of the slide. And what is very clear is that the luculi, so the gel area of the tomato, are almost anoxic. And this is not too surprising because it's a gel. There are no pores inside the gel, so the diffusivity of oxygen is very low. It's basically the diffusivity of oxygen in water. So there's almost no transport of oxygen, and yet there is metabolic activity, so all the oxygen is basically consumed. But what we can also see is that in the unripe tomato fruit, so the mature green and the orange one, everything is kind of bluish, which means that there's an hypoxic situation in the fruit during the ripening process, which is alleviated when the tomato moves on to the red ripe phase. So the color is more reddish, which means a higher oxygen. And at this stage, the ripening program is declining. So ethylene production, respiration declines. And because there's less oxygen need, the oxygen concentration increases. What is also very interesting is that there's a red zone here at the pedicel scar, so where the abscission zone basically, which suggests that this is a major transport pathway of oxygen inside the fruit, and there's hardly anything coming in from through the epidermis and the cuticle. So we decided to investigate this in more detail, and we did an experiment where we covered the pedicel with Vaseline and another one where we covered the cuticle with Vaseline. And here in the y-axis, you can see the respiration rate, and you can see that the respiration rate when the pedicel scar is blocked is much lower, while blocking the cuticle with Vaseline does not seem to have too much effect. This again seems to indicate that all the oxygen that comes in the fruit has to come through the pedicel scar. And if you block it, there's just no oxygen. And if there's no oxygen, there's also not going to be too much respiration. And we then looked at the microstructure of the pedicel scar. And for this, we excised the pedicel scar and put it in the X-ray micro CT machine, and we visualized the structure. And this is basically the scar. So this is a contrast between air pores in black and the cells in gray. And you can see that if you that there's a lot of black inside, which means that there are many air pores in it. And if we did when we did a 3D visualization, we found that this pedicel scar area is basically very porous. It has a porosity of almost 70%. And this is the reason why oxygen can diffuse so fast through the pedicel scar. And then finally, we validated the model with oxygen sensors together with Ole Pedersen's lab, University of Copenhagen in Denmark. And here you can see an oxygen sensor that was inserted at various positions inside the tomato tissue. And the blue dots represent the oxygen concentration measurements. And the red line is the model simulation. And you can see that in this red tomato, the oxygen concentration is relatively high, almost like 17%, 18%. But then if we reach the locali, then the oxygen concentration drops to around 3 to 4%, and then it increases again when the needle reaches, or the sensor needle reaches the columella, towards the center of the fruit. So this kind of showed us that our model was accurate and reliable, and that we can use it for further research. To conclude, the pedicel scar is the main gas diffusion channel during ripening. Gas transport model is validated. The gel is hypoxic, and this is because of the high oxygen consumption and the diffusive resistance of the water in the gel. And then we are now investigating the effect of oxygen and carbon dioxide on ethylene biosynthesis and cycling, because it is very interesting that the development of these hypoxic zones in the pericarp kind of coincides with the climacteric rise, which sort of seems to indicate that there also might be kind of a role of hypoxia in the development of tomato fruits. And with this, I would like to thank the rest of my lab for helping me out and doing all this nice research, and also you to listen to me. Thank you.
Thank you very much, Bart. It was really interesting, very cool tools you have there. And we already have some questions. So our anonymous attendee asked, does ripening can be delayed if a pedicel scar is removed?
I'm not sure how to answer that because, for example, the difference between tomato harvest in Belgium and in the United States is that we basically cut the pedicel rather than removing the tomato from the pedicel. So, but if you remove the tomato at the pedicel, then you have this scar, and I'm not sure what you mean with removing it. Is that excising the pedicel? I think that if you block the pedicel, for example, after harvest, there could be some advantage in the sense that if you hinder the oxygen transport through the pedicel scar by blocking it, then as oxygen is a substrate for ethylene biosynthesis, you might also retard the ethylene biosynthesis and in this case, the ripening of the tomato. So I'm not sure whether this is an answer, but this is what I think you could do.
Another question asked by Seila Zellman is whether the post-harvest oxygenation can be affected by ambient humidity.
No, humidity is always an issue in post-harvest storage because typically fruit or vegetables would lose water during post-harvest storage. But there is not a direct connection, except the fact that, of course, water is an end product of respiration. So if you would manipulate the oxygen concentration, then in a way you manipulate the respiration metabolism and also the water production because of the respiration. And this might have some effect on water loss to the environment. But I think this is rather minimal.
And we have time for one last question. Rory Osborne asked, do you think that the hypoxic environment within the tomato fruit is essential for seed development, or is it an evolutionary accident due to the high respiration rate required to produce seed?
That's a very interesting question. I can't answer that, but this is what we are really interested. So at this point, we just know that if you look at, we're actually also modeling the ethylene biosynthesis, and oxygen is, as I mentioned before, a substrate for ethylene biosynthesis. So if there's not too much oxygen available, you would expect lower ethylene biosynthesis and also lower ethylene signaling and so on. So it is very interesting to think about the consequences of that, and definitely there might be a role of hypoxia, a direct role of hypoxia into the development of the seeds.
Thank you. There are many more questions. You can see them in the Q&A box, and you can type the answers directly to them. Because now, thank you again, it's time to move to our second speaker, Amanda Cardoso, who's an assistant professor of crop physiology at North Carolina State University. Her studies focus on identifying climate-resilient crops, uncovering structural and functional resilience mechanisms, and generating knowledge on best management practices for plant production under challenging climates. Today, her talk will focus on her recent paper on Phaseolus vulgaris impairments in response to waterlogging. Looking forward to your talk, Amanda.
Thank you. Can you see my slides?
Yes.
Okay. So thank you, Alicia, for the introduction, and thank you, Mary, and all the organizers for the invitation to be here today talking about my research. I'm going to briefly introduce this study that we performed in my lab that investigated plant hydraulic impairments in response to waterlogging. So first, I would like to describe what plant hydraulic means. And in this context, it refers to the transport of water from the soils being absorbed by the roots, transported to the shoot, and then being lost by the leaves through transpiration. So the efficiency of the water transport within the plant body is what we refer to when we say plant hydraulics. And the hydraulic conductivity or the efficiency of the water transport of the plants depends on the efficiency of the water transport in roots, stems, and leaves. and the damage or reduction to the water transport in any of these organs is going to ultimately affect the water transport in the whole plant. So the vast majority of studies investigating plant hydraulic impairments are done in response to drought. We know that during drought, leaves, roots, and stems will have declines in the hydraulic conductance as the plants dehydrate. So in here, we're talking about plant dehydration, and this is being described by declines in water potential. So as the water potential gets more negative, then we have a decline in the hydraulic conductance of stems, roots, and leaves. And this data is shown for 3 species, but this is a common knowledge for us working in drought, that these impairments to the plant hydraulics happens during drought. Now, there's a very little understanding of what happens during waterlogging, and that's why I decided to investigate that. And before I move to the next slide, I would like to explain a little bit the causes of plant leaf hydraulic impairments and root hydraulic impairments during as the plants or organs desiccate. And in these organs, we know that aquaporins are deactivated during drought, which increases the resistance of water transport in cell membranes. There's also xylem embolism occurring. Xylem is the tissue responsible for long distance transport of water in plants. And during drought or desiccation, the xylem is going to absorb or air bubbles will enter the xylem and will create an air cavity that prevents the water transport in that xylem conduit. So both of these are causes of declines in hydraulic conductance of leaves and roots during drought. And in stems, the declines in hydraulic conductance are mostly associated with embolism. Now, what we know that happens during waterlogging, and this is very nicely described in the literature, is that the roots are very impacted. And the water transporting roots are impacted during soil waterlogging. And this is because of the lack of oxygen in the soil, and all the biochemical changes that occur in root cells will lead to declines in the root hydraulic conductance, described in here as K root. And this will limit the ability of plants to uptake water from the soils and transport water from the roots to the shoot. And so we know that during waterlogging, plants will dehydrate, and we can visualize wilting of the leaves. So this is pretty much well described in the literature. And during the soil drainage after the waterlogging, roots continue to have a very low hydraulic conductance, especially in sensitive plants, because of a massive production of reactive oxygen species. And the shoot can dehydrate even more, and we can see mortality of leaves, and eventually in very sensitive plants, the whole plant mortality. So this is the extent of what we know happens in terms of hydraulic impairments during waterlogging. And we decided to go further and investigate what happens to the shoot in terms of water limitations to water transport. For that, we selected Phaseolus vulgaris as a model species because it's a very sensitive legume crop. And the first question that we asked was if leaves would also suffer hydraulic impairments in response to waterlogging, in a similar way to what happens to roots. And we saw that it does. So this plot shows leaf hydraulic conductance measured for control plants and for waterlogged plants during 72 hours of waterlogging. and during 48 hours after waterlogging. And we see a massive decline that occurs within 24 hours in the ability of leaves to transport water when the roots are waterlogged. So about 50% to 55% decline in leaf hydraulic conductance, which continues throughout the extent of the waterlogging period, and it does not recover very fast. So during 48 hours, the leaves do not regain that ability to transport water. So the first answer is yes, in the sensitive species, leaves do suffer hydraulic impairments during and after waterlogging. Now, one question that we had was, because leaves, when they dehydrate, they lose the ability to transport water, and we have two main causes, embolism or aquaporins, we wondered if that would be a result of xylem embolism, which is something that we can accurately measure in our lab. So we measured the accumulation of embolism in the xylem of the leaves in vivo as the plants were subjected to 72 hours of waterlogging and then 48 hours of post-waterlogging. And we saw that the majority of embolism, so we can see increases in embolism accumulation in leaves, occurs after the waterlogging. And we have very little embolism during waterlogging. And if you remember from my previous image, we had 55% decline in leaf hydraulic conductance by 24 hours when there's absolutely no embolism, and almost all leaf hydraulic impairment occurred by 48 hours of waterlogging when again there's no embolism. So the answer was that no, the declines in leaf hydraulic conductance preceded the formation of embolism. So they're likely linked to aquaporin dysfunction, which is something that we still have to measure, but we couldn't measure for this particular study, but it's something to be proven. Okay. Irrespective of the cause, we wanted to know if what was driven the leaf hydraulic impairment was simply a function of shoot dehydration because of the roots not being able to absorb and transport water, or if there was any other potential biochemical alteration in leaves in a similar way to what occurs in roots, and that would lead to this impairment. And what we saw was that the way that we had to do to prove that was that we had to plot our waterlogging data. So in here we have the leaf hydraulic conductance observed before waterlogging, after 24, 48, and 72 hours, and during the post-waterlogging. So we have these data, and we know the minimum water potential, or the minimum or the maximum dehydration that those leaves experience, and at the time that they showed that hydraulic impairment. And we plotted that against data that we had of leaf hydraulic impairments occurring during drought. So the gray symbols and curves are what we observed during drought, when dehydration alone can explain this leaf hydraulic impairments. And we see that the majority of the data overlaps. So there is a big component or a big overlap between drought and waterlogging when it comes to limiting the ability of the shoots to transport water, specifically the leaves, but very early on during waterlogging. So within 24 hours, we had, again, a massive decline in the ability of leaves to transport water that could not be explained by dehydration alone. So this point that should be around here, anywhere between here, it's much lower, a much lower position. So only 12% of the decline in leaf hydraulic conductance could be explained by dehydration, and we actually saw 55% decline. So there is something else going on in the leaves that also need to be further studied, and we believe that chemical signals, potentially the same ones limiting stomatal closure, stomatal opening, during waterlogging are also limiting the ability of the leaves to transport water because of alterations in aquaporins. So just some conclusions. In addition to roots, leaves are also hydraulically impaired during waterlogging and after waterlogging as well. That impairment continues. The hydraulic impairments in leaves during waterlogging are not the direct result of embolism formation. So there's likely an aquaporin dysfunction occurring. And the hydraulic impairments in leaves during waterlogging cannot be fully explained by the dehydration of those leaves. So we believe that there are chemical signals that are limiting aquaporin function and thus water transport in leaves. So this is just a snapshot of like our study, and we're just covering a little bit of that study. There's much more, and if you want to understand everything about water transport during waterlogging that we did in this paper, you can see the full paper, the link to the full paper here. And I would like to thank my postdoc, Eduardo, who was the first author. So here we have him performing the waterlogging experiments, and we writing the paper or going over the paper during a very long road trip that we took to Purdue University. And thank you all for your attention as well.
Thank you, Amanda. That was great. Very insightful talk. And actually, there are already many questions, but I think we have time for one or two. So Elisa Montruccio asked, is there a difference in the leaf hydraulic conductance between young and old leaf in control conditions and in the waterlogging?
Yes, great question. Yes, we know that the ability of leaves to transport water declines as they age. And this is something that we briefly discussed in the paper, I believe, is that the lower leaves, the leaves in the lower canopy that are older, they suffer much more intense hydraulic limitations during waterlogging than the upper canopy. So there is a canopy differentiation in terms of the damage, and that is related to leaf age. Yeah.
So before I ask another question, could you please return to the QR code for your paper because someone didn't catch it in time?
Oh, yes. Let me go back here. Yeah.
So the last question asked by Sean Hartman, are the xylem embolisms truly happening because of reoxygenation? Or could it be that there is a lag phase of waterlogging damage that you only observe at this time? So in other words, if you waterlogged longer, would you also see embolism after like 5 days?
Great question. This is something that we have to test. And we don't know if it is, what we believe is that it's just an extension of all the root damage occurring during waterlogging when they're fully exposed to all that oxygen. Biochemically, they cannot cope with that oxygen. Their impairments in uptaking water and transporting to the shoot are even more impaired. And so the shoot dehydrates more. So it is a reflex. What we believe it is a reflection of the ability of the shoot to receive water from the roots.
Thank you. Thank you, Amanda. I also invite you now to the Q&A box. You can type directly the answer. There are many, many more questions.
Thank you.
And now I would like to move to our next speaker, Moto Ashikari, a professor at the Bioscience and Biotechnology Center in Nagoya University. His lab is researching various useful agricultural traits in rice. His lab has identified several genes and genetic pathways that are involved in rice growth and yield, as well as responses to flooding. The lab also explores genetic diversity in wild rice relatives and has identified genes that have been altered during domestication. His research is contributing to breeding projects to improve rice in Asia and Africa. And today he will explain us the different strategies of rice to cope with submergence. The floor is yours, Moto.
Yeah, thank you very much. Can I share my slide?
For now, we don't see them yet.
Okay.
Yes, perfect.
Okay. Oh, sure. Okay. So thank you very much for invitation. My name is Motoyuki Ashikari, in Nagoya University in Japan. Today, I want to introduce our review paper in Plant Physiology. Title is Escape and Quiescence Strategies of Lice to Cope with Submergence. This review paper is written by me, Moto, with Kay and Julia Bailey-Serres. Okay, so lice is one of the most important crops for humans. Lice provided about 20% calorie human consumes. And many countries produce lice, but almost 90% of lice produced and consumed in Asia. So lice is very important food for our meals. So this is this picture shows that the average of annual precipitation in the world. Here is Asia. So Asia belonging to the monsoon climates. This monsoon climate is suitable for the rice productions. So this is a paddy field in our university. You can see the many rice seedlings in the paddy field. So rice can grow and in this kind of the waterlogging conditions. However, other many cereals such as wheat, barley, corn cannot grow well in this condition. Why can rice grow in this waterlogging conditions? This is a structure of rice. So rice has a leaf, erythema in leaves, and the erythema in the leaf sheaths. Yes, and the erythema in the stem, and the erythema in the roots. So this connecting this erythema helps this bleed. Okay, so this connecting this whole structure helps breathing under the partial semi-submergence. Okay, so monsoon climate in Asia has a dry season and a rainy season. So here is Thailand. Okay, this is a picture taken by satellite in dry season and the rainy season at the same point. You can see the difference of water volume on the fields. Okay, so heavy rain can cause flooding. So in Asia, we have the two kinds of the flooding. One is flash flooding. The other is deep water flooding. Okay, so some of lights has a strategy against for flash flooding called quiescence strategies. The other lice plants has surviving strategies against the deep water flooding. That's called escape strategies. Firstly, I want to show that the quiescent strategy against the flash flooding. Okay, so flash flooding, the water levels rise rapidly at the lice seeding stage, and the lice is submerged for a few weeks. This time, lice plant try to elongate, avoid hypoxia conditions, and consume the energy a lot. After the water reduced, and the lice can not grow anymore because depletion of the energies. So, however, this is the wild type plants, so intolerance to flash floodings. So after the 14 days submergence, the plants cannot grow anymore. However, plants having the submergence encoded AP2 transcription factors can allow growing after the submergence. So this submergence has identified David McGill group and Julia's groups. Okay, and generalized culture consume the energy under the waters. However, plants having the submergence under the waters, so they do not elongate and then keep the energy as a quiescent state. So after reducing the water, the plant regrows using the saved energies. That is a mechanism. Okay. And the molecular mechanism of this survival strategies against the flash flood was many analyzed by the researchers. Okay, in the waters, rice plants accumulates ethylene gas, one of the plant hormones, because of the production of the ethylene gas and the low diffusion in the waters. So accumulated ethylene gas phosphorylates sub-1A proteins and then signaling block gibberellins and the branchinus neuron and so on. And also activates anaerobic survival genes, fermentation genes. So due to the limitation today, I do not explain details, but if you're interested in, please visit our review papers. Okay, so this is a bleeding story about sub-1 genes. This is Swarna, such as elite variety Indian regions. And this is sub-1 donors. Bleeders international research institutions shall transfer these genes by backcrossing with Markus selections. Finally, they produce Swarna sub-1 subvarieties. So backgrounds of the genetic backgrounds are Swarnas. However, the Swarna sub-1 possesses positive sub-1 genes. And this line is already spread in the commercially available in the Southeast Asia now. So this is maybe a good example. The basic research can be used in the social implementation. Okay, so let's move to the next topics. So escape strategy against the deep water flooding. Okay, so deep water flooding, the water level slowly rises a few meters, and it contains for a few months, continue a few months. So in case of the generalized cultivar, usually the generalized cultivar planted in the shallow water condition or dry conditions. Once in deep water condition, plant cannot breathe and then drown and die soon. Okay. So in case of deep water rice, when deep water rice planted in the shallow water conditions, plant feature is very similar to the general rice cultivar. However, once in flat conditions, deep water rice can elongate according to the water depths. Okay, so this is the movie escape strategy of the deep water rice by stem elongations. Left plants is the general rice cultivars. Right plants is the deep water rice. Okay. Okay, you can see the movies. So left plants general cultivar cannot elongate in the waters. However, deep water rice can elongate in the waters and then catch the oxygen from the top of the leaves above the water surface. Okay, so next movie is you can feel that the speed of elongation of the stem. Tank was filled to the brim the first day. Okay, so deep water rice elongates the stem dramatically, about 20-25 centimeters a day. Such amazing elongations to catch the oxygen above the surface of the waters. Okay, so this is a stem structure and its elongation. Okay, so in air conditions, stem that means it's composed of the node, internode is packed in the stems. However, in the deep water conditions, intercalary meristem located between the node and the node starts the cell propagation, and then cell elongation occurs. Okay. So this is another movie. Okay, so this is a leaf and the node in the internals. We mark this every 5 milliliters by black pens. Okay, and then so here is the movie. So please focus about this 5 milliliters because intercalary meristem located this 5 milliliters. Okay, shows that so dramatic cell propagation. Cell elongation is occur at this 5 milliliters. So and then rice stem elongates dramatically. So that is amazing, right? Okay, so how long can deep water rice elongate? So in the air conditions, rice usually 1 meters. However, deep water rice can elongate dramatically around 5-6 meters. And here is panicus. Yes. Okay. So Generalized cultivar cannot elongate in the waters. However, deep water lice can elongate. What is the difference of the molecular levels and the physiological levels? So firstly, we doubt this is plant hormone gibberellin because gibberellin is associated with stem elongations. So I skip over the detail. However, we identified that the deep water lice produces gibberellin for under the waters. However, generalized cannot produce the amount of the gibberellins in the water. This is one difference between generalized and the deep water lice. And we also doubt the gibberellin signal on the gibberellin responsivity between them. Okay, so we tested the gibberellin sensitivity between the generalized cultivar and the deep water lice. So firstly, we apply the uniconazole. That uniconazole is the GA biosynthetic inhibitor. And then we apply the gibberellin at the different levels. Okay, and then we grow the 3 weeks, and then so we measure plant heights and stem lengths. Okay, this is the data of generalized cultivars. When we apply the uniconazole, plant becomes shorter. This is reasonable because uniconazole is a blocker of the gibberellin biosynthesis. And then in this condition, we apply the gibberellin as the plant's height becomes taller and taller according to the gibberellin levels. Such is reasonable, okay? And this is the deep water lice. When we apply the uniconazole, plant becomes shorter and the plant heights become longer and longer. Looks like generalized cultivars. So leaf elongates GA-dependent manner in T65 and the C9085, just a deep water lice. Okay. However, in the case of stem, okay, so this is the data of generalized cultivar. When we apply the gibberellins at high concentrations, however, generalized never elongates the stem. In the case of deep water lice, the white triangle indicates a node. Between node and the nodes are internodes. Okay, so deep water can respond to GA and then stem elongation occurs. So internode of deep water lice respond to GA. However, generalized cultivar cannot show response to GA in stem. So both of leaf can respond to GA as the same manner. However, gibberellin responsivity in the stem is different. Okay, so this is left is the generalized cultivar and right is the deep water lice. And the yellow bar indicates the chromosome of the generalized cultivar, and the blue bar indicates the chromosome of deep water lice. So I skip over the genetic data. However, we found the 4 major QTL in the deep water lice. Okay, among 4 QTL, chromosome 3 and chromosome 12 QTL is work as gibberellin responsivity. Okay, so we try to identify these genes, and then finally we found ACE1 such as a peptide, and then so another gene is DEC1. So also this gene is identified Fabio's group in the Italy. So this gene encoding C282 transcription factors. Okay, so I can due to the time limitation, I skip all the details. However, I want to summarize the mechanism. Okay, ACE1 that is an activator of stem elongation. Probably this ACE1 activates intercalary meristem. However, DEC1 that is a suppressor of stem elongation. So ACE1 and the DEC1 work as an antagonistically. Okay, so I'm sorry, I skip same. Okay, so we also identify the other QTL genes. So that is chromosome 1 QTL encodes the GA biosynthetic genes, and then another QTL encodes so AP2 transcription factors. Okay, so now we this is a current model of internodal elongation in deep water lice. Okay, the one that is a suppressor of internodal elongation is expressed in the air conditions. However, decline expression is downregulated by the submergence or GAs. And then the plants express the GA20 oxidase too. That is the gibberellin biosensitive genes produces the gibberellins under the waters. And then the ACE1 also expressed by the submergence. ACE1 is activators. And also SK1, SK2, another transcription factor, enhances stem elongations. Okay, so now is 4 factors is involving of stem elongation in the water in deep water lice. So now we are trying to find out the other molecules regulated of this mechanism. Okay. Now it's most of the time. Okay, so here today I want to introduce 2 types of strategy against flooding in lice. One is quiescence strategy for flash flooding. The other is escape strategy against for the deep water flooding. So if you're interested in more, please visit our review paper. Thank you very much for attention today.
Thank you, Moto. Every time I think we know everything about rice and flooding, your group surprises me. Really great talk.
Yeah, thank you very much.
We are a bit out of the time, but just one question.
Okay.
Ashim Kumar Das asked, with the help of escape strategy, few Korean rices can move upward to get oxygen. But I found that under salt and submergence together, they cannot move upward. When coastal flooding increases rapidly, how SAP1 or SK12 or ACE1 DEC1 can work under both salt submerged conditions?
Okay, that is interesting, but honestly, I have not tested the combination of the SAP1 genes and the other deep water lice genes. And so if we combine, I don't know what phenomena happens. Yes. So yeah, we want to try. Yeah. So, but at this moment, I don't know that's what happens. And also interested in this phenomena he or she found. Yeah, I'm very interested in. Thank you.
Thank you. Unfortunately, we have to move on to our next speaker, but also please check the Q&A box. There are more questions and you can type the answers to them. Thank you very much.
Thank you.
And our final speaker is Beatrice Giuntoli, an associate professor of plant physiology in the University of Pisa. Beatrice has investigated the physiological consequences of hypoxia and oxygen fluctuation in plants, contributing to the discovery of direct oxygen sensing mechanism based on cysteine oxidase enzymes. Her research is focused on the design of genetically encoded biosensors in plants, the reconstruction of synthetic oxygen-regulated mechanism in eukaryotes, and the development of optogenetic tools for plants and bacteria. And today she will guide us through the synthetic biology strategies to investigate hypoxia. We are eager to hear from you, Bea.
Thank you, Alan, for the introduction, and thank you for inviting me today. Sorry, my voice is not at the best today, but I hope it's fair anyway. Okay, I think you can see my screen. Okay.
Yes, everything fine.
Okay, so I'll start to tell you something about synthetic biology applications in plant hypoxia biology today. So we have heard from previous speakers that hypoxia can associate to plant tissues for several reasons, so I will not spend much words about it, but you can find hypoxic niches in plants due to physiological reasons. You can have hypoxia as a stress, and you can have hypoxia in response to some biotic interactions in plants. So how can synthetic biology help the study of hypoxia biology? I think we can find at least 3 fields of application. So in the first case, synthetic biology can help to design and test reporters, so genetically encoded biosensors, that can help us monitor the occurrence of hypoxia in plant tissues. As Rashmi mentioned, a number of studies have allowed us to spot hypoxia inside plant tissues thanks to the use of genetically encoded reporters, as we can see from this nice schematic prepared by Dan in the past. All the reporters that we used so far, the genetically encoded ones, are based on the activity of hypoxia signaling. So it means that they tell us that hypoxia signaling is ongoing. They don't tell us exactly the level of oxygen in the tissues. So this is why I would say one of the hot points in synthetic biology of plant hypoxia is looking for reporters that are able to respond directly to oxygen variations, so for direct biosensing. So far, we have candidate proteins to work like these that are fluorescent proteins. So synthetic biology can help us, I would say, in choosing the right fluorophores and testing the fluorophores once expressed in plants and improve them for this application. We can have use synthetic biology to engineer responses to oxygen in a precise way. So we believe that we can exploit oxygen responsive mechanisms from foreign organisms like animals, for instance. So extract these the components of these mechanisms, express them in plants where they're meant to crosstalk the least with endogenous responses. This feature of low crosstalk is also called orthogonality, using a synthetic biology term. So we believe that engineering these kind of switches, genetic switches, can enable us to control better oxygen responses in the transgenic plants. And the third field of application of synthetic biology that I may propose is that it can help us actually to study how oxygen responsive mechanisms from plants work. So we can think of identifying the components of oxygen responses in plants, extract them from plants and express them, reassemble them in different organisms that are more tractable, more easy to study. And these new host organisms can give us answers about oxygen responses do work in the original plant hosts. So due to time limitations, I think I will give you only examples of these third application that are direct experience coming from my lab. I would like to point out that, okay, there are limitations, in my opinion, in the use of synthetic biology for hypoxia research. These limitations are corresponding to the bottlenecks that the synthetic biology is facing in plants at all nowadays. So synthetic biology offers a workflow if you want to engineer genetic circuits. So first you start designing the DNA circuit, then you assemble physically the DNA circuit and express it in a host where you can test how the circuit behaves, and from the outcomes of the test, learn how to improve your initial circuits, and then a second cycle of DBTL operation can start. So I would say that nowadays, plant synthetic biology suffers from limitations, mostly in the design and test part. So what we lack the most is the catalog of plant DNA parts, means promoters, coding sequences, terminators, other regulatory sequences, that we know exactly how they work. So standardized parts to be used to assemble our circuits. And another main limitation is that there is limited choice of the test beds where to see how our circuits work. So, of course, we can go for heterologous test beds. The plant systems that we use so far are fantastic, I would say. They are mainly isolated protoplasts or Nicotiana benthamiana leaves. The problem is that these are transient systems and they may not mirror what happens then in the transgenic plant when the circuit is introduced stably. Okay, so I would focus now for the rest of the talk on the main application that we made of synthetic biology in my lab for hypoxia research. So we tried to understand better how a pathway from plants work. Is a pathway of paramount interest for us because it's the pathway for oxygen sensing, called cysteine and diagram pathway that you can see here. So what we tried to do, as I said, was abstracting the components of this pathway from plants, expressing them in another host organism, which is yeast, to understand how the pathway works. Very schematically, the cysteine and degron pathway is dedicated to the degradation of some proteins through the proteasome when oxygen is present. The most important proteins processed are these transcription factors, ERF7 factors. When oxygen is there, they expose an N-terminal cysteine that can be oxidized by the oxygen sensors of plants that are plant cysteine oxidases, these PCOs. And once oxidized, the ERF7 proteins can be processed subsequently by arginine transferases that conjugate arginine and recognized by E3 ligases like PRT6 that conjugate ubiquitin and degrade, led to degradation of the protein. When oxygen is not there, PCOs are disabled, so the ERF7 proteins are stable and they can go to the nucleus and activate the transcription of hypoxia responsive genes. So this is the pathway that we played around. What we did was reconstructing this pathway fully in yeast cells. I'll tell you in a minute why yeast. So once we have our synthetic cysteine and degron pathway in yeast, since the pathway is modular, so it's made of steps, it's very versatile. So we can use the yeast to interrogate the pathway. So we can replace the cysteine oxidases to test candidates. We can replace the final substrate proteins to study them. We can treat yeast with chemicals to understand if these chemicals can modulate the activity of the pathway. So the way it works in yeast is like this. We chose yeast because it has a similar degradation pathway for proteins than the one I described. So yeast is able to take proteins starting with aspartate or arginine or glutamate, sorry, and degrade them through the proteasome thanks to ATE and E3 ligase. What yeast cannot do naturally is processing cysteine starting proteins. And this is because it lacks cysteine oxidases. So all we had to do in yeast was expressing a plant cysteine oxidase from plants to enable a synthetic cysteine and degron pathway. And since yeast uses completely different mechanisms for oxygen sensing, we believe that this pathway is orthogonal. To see if how the pathway is working, we introduced a visual substrate for degradation that you can see here. The important thing is that this substrate will release a portion protein that exposes a cysteine and is conjugating to firefly luciferase. So measuring the amount of luciferase activity, we can understand the stability of this protein. And then there is another part of the protein which is insensitive to oxygen that we can use for signal normalization. So the first thing we did at that time was checking that this substrate responds to differences in the availability of atmospheric oxygen to yeast cultures. And as you can see here, the substrate becomes more and more stabilized when oxygen falls below 5% in the atmosphere around. We expected this kind of response, and we were very happy to see it at the beginning. So I'll show you two applications of these synthetic cysteine and degron pathway that we made so far. It's not everything that we did, but I'll curtail our excitation and experiments today. So in one of our approaches, we tested chemicals to find chemicals that are able to modulate the cysteine and degron pathway because we believe that they can be of agronomical interest. And particularly, we looked for inhibitors of the cysteine and degron pathway that can be in the future used to prime plants before hypoxic stresses come, like submergence or waterlogging. So as you can see here, we probed a library of around 2,000 chemicals. And with our approach, with our synthetic strain, we could narrow down this number to 3 hypoxia mimetic molecules that you can see on the right here that can be used for subsequent chemical improvement. In our hands, these 3 molecules are genuine inhibitors of the PCO enzymes. And this is because they only stabilize our synthetic substrate if the substrate is starting with a cysteine, but they cannot stabilize substrates that are starting with aspartate or arginine that will be processed by downstream enzymes of the cysteine and degron pathway. And the other application that we are finalizing now of this synthetic cysteine endogenous pathway yeast strain is the search for novel protein substrates of the cysteine endogenous pathway from the proteome of a plant, which is Arabidopsis. Because the interesting point is that if you look in the proteome of Arabidopsis, there is a vast number of proteins that have cysteine in second position, but only a handful of them have been proven as proper substrates of the cysteine endogenous pathway, only 7. So what we did was replacing, our synthetic substrates that initially contained the N-terminal portion of an ERF7 protein with substrates that contain the N-terminal portion of other candidates from the Arabidopsis proteome. So we did it for 61 new proteins from the proteome, and we checked the stability of these novel substrates when the pathway is enabled in yeast or when the pathway is disabled, like under hypoxia or in the absence of the enzyme. So in this way, we could identify so far 15 novel candidates from the proteome of the cysteine endogenous pathway of regulation, implying that this pathway can impact on plant physiology way more than we have expected so far. So with this brief talk, I think at least I could convince you that synthetic biology can be of aid in the study of plant hypoxia. And I would like to thank you all and all the people that contributed to this research in the past years, starting from people from PISA, so from UniP, and from the Plant Lab of Sant'Anna, in particular, Michela Villapuerta and Noemi Lamonaca, who worked on yeast in these years. I would like to thank collaborators from the University of Oxford, a number of them, and collaborators from other institutions from PISA and from Spain. Thank you.
Thank you, Bea. Very complicated topic, but presented in a very clear way. So maybe I will start with a question. So every time I hear you talk about the synthetic biology, it seems that only the sky is the limit with what we can achieve. In your opinion, what is the next big step in hypoxic studies when it comes to synthetic biology?
Next step, I can't predict, maybe. I can tell you like two opinions on two matters. So one big thing that we would like to achieve with synthetic biology would be exploring the functional space of plant cysteine oxidases. Meaning, I believe that this gives us the opportunity of testing these kind of strategies. Of testing a number of variants, also random variants of PCO enzymes. We can apply directed evolution to yeast quite easily. So we are confident that we can explore really the functionality of the plant cysteine oxidases by mutagenesis and directed evolution, and tell people more about what it takes to be a tile processing enzyme, an oxygen sensor, and so on. And the second, challenge that I see instead is connected to a limitation, I would say, of plant synthetic biology. So now we're mostly going with a one-by-one approach, one gene by one approach. So if we engineer genetic devices in using synthetic biology, it's very likely that we are focusing on one regulator and try to introduce it in the plant and see how we can improve it. But the responses to hypoxia are quantitative. So we need to find a way to make synthetic biology useful, I think, also in this case of working with quantitative traits.
Thank you. And in your experience, how well what you find in yeast with your tools translates to what happens in plants? Like, are you successful in confirming, because at the end, those are very different organisms. So I guess this is one of the other challenges that you're getting during your work.
So thank you for the second question. Very briefly, we are doing this. We are then proceeding to the confirmation of our results in plants. This we are doing, for instance, for the with the substrates. So novel substrates are going to be, of course, confirmed in plants. What I can say is that actually this strategy of abstracting to another organism can become particularly valuable when we compare the differences between the new model organism and the original organism. So this difference can tell us about the additional functions that we didn't know in the original organisms, that the pathway, for instance, is carried out.
Okay, thank you very much.
Thank you.
And we have many questions, but sometimes they are still coming for the previous talk, so I have to understand for who are they. Okay, I think that's gonna be it for now. If there are any more questions, then please feel free to contact Beatrice personally, or by email, I mean. And I would like to again thank all of our speakers. It was really great lineup. We had a chance to see hypoxic research taken from very different approaches using many different plants. Thank you everyone for attending. We had a very great number of people in the audience. And I think the next webinar that Plant Physiology is organizing is in February on lipids. That's right,
Yes.
Mary?
You stole my line. Yeah, well, first of all,
sorry.
thank you, Bart, Amanda, Moto, and Beatrice. Those were really exciting talks. I'm so glad you cheered me up. And Alicia, wonderful job. Thank you. Attendees, please see my note about getting your certificate of attendance if you need one. An email will come tomorrow. And as Alicia said, we've got a webinar in February on lipids. This is the February focus issue of Plant Physiology. I think it's February 13th. We've sent it out by social, and check the website. Once again, thank you very much, speakers. Those were fabulous talks. I was very excited listening to you. And thank you, Alicia. Thank you, attendees. See you next time. Bye-bye.
Bye-bye. Thank you.